For our project, we decided to look into if mice in the United States outlive mice from other countries.
Introduction of urbanization. Severe amounts of environmental change can cause species to adapt quickly, and some species that have grown the best at this are rodents. Rodents, like rats and mice, can be found in all kinds of environments, ranging from rural to urban. They also seem to become riskier and are more willing to explore in more urbanized environments. Urban areas have a population of 50,000 or more. Urban clusters have a population between 2,500 and 50,000. So when a country has higher populations and in concentrated areas, this will bring about riskier and bolder movements of the rodents. This can be a feature that actually helps them survive longer, as discussed later in the presentation.
Anatomy of the house mouse. Body structure of mice is a long body that thins at the head. They have 4 legs and a long tail. House mice have vibrissae, or their whiskers, for detecting objects around them and navigate dark environments, which helps the mice because they are nocturnal. They are omnivorous. They eat seeds, grains, grasses, and some small invertebrates, as well as things they can gather and hold. All these features allow mice to be able to explore more areas even in less than ideal conditions such as darkness. Since they can explore more and eat a wider variety of foods, this means they will be very likely to survive in any environment they find themselves in.
Factors that determine age. Their survival capabilities are dependent on their age as well as urbanization. Exploring and motor activity peak around young adulthood and start slowly declining in mature adulthood. Boldness is an important factor that depends on the organism's ability to determine what risks are worth taking for survival. This factor diminishes with age. Though mice do not tend to live long due to these listed factors, with a higher level of urbanization leads to the increased availability of resources to the mice.
Where house mice reside. Due to human aid, house mice have been able to adapt to a variety of habitats all over the world, taking refuge in all the continents in present day. Since house mice are adapted well to wherever they reside in current day, they would likely survive longer where there is more urbanization, which coincides with larger populations. As mentioned, with higher amounts of humans comes the increased availability of shelter and food for these organisms. With more urbanization comes more humans, and more humans brings the ability for the mice to live. Due to their adaptation to all environments, it is likely that the climate or terrain of the environment may not matter as much as the presence or lack thereof of humans.
So for our project, our research question was, do mice in the United States outlive mice from other countries? Our hypothesis was the United States has the highest population and therefore the highest level of urbanization. Out of the other countries it'll be compared to, so house mice will likely live longer in the United States than in the other countries because of the more widespread urbanization and the higher likelihood of human aid for survival.
For our methods, we use the link in the PowerPoint to collect our data for Mus musculus by typing in the genus and species, and then we selected the length, mass, sex, and life stage to be included. The data was then placed into an Excel spreadsheet, and the museum samples were ordered by country, and the value of life stages were converted to the established keys of juvenile, young adult, mature adult, late adult, old, and senescent. This attempted to keep accuracy by allowing all the data to function through the same values rather than how they first appeared in the original data, and the life stages were based on a study of house mice done by Radulesk and other researchers.
The 4 countries with the most data besides the United States were separated into their own sheets. These included Panama, Ecuador, Canada, and Russia. Each sheet contained all of the life stages of the mice, along with a column of the United States data which was used for analysis. Using a NOVA single-factor test for analysis, all these countries' data were compared to the United States data individually to find the p-values. A p-value that is less than 0.05 is known as significant.
For our results, the average mouse life stage was found for the United States. It was 2.758. For Panama, it was 2.881. Ecuador, 2.410. Canada, 2.725. And Russia, 2.313. All of the p-values were found in comparison to the United States. The only one that wasn't significant was Canada, and due to Panama having a higher significant life stage than the US, and Canada not having a significant difference compared to the US suggests that urbanization is not the main factor for house mice living longer.
For our discussion, we found that the difference between house mice that live in Canada and the United States were found to be almost the same, not having any significant difference. According to the results, the p-value between the United States and Canada was found to be 0.993948 and was considered insignificant. This suggests that urbanization is not the determining factor for mice to live longer. This could possibly be because all the samples from Canada being found in the same area around Edmonton, which is a highly populated city in Canada. In comparison, the other countries have samples from multiple places around the country rather than one city.
The other three countries, Ecuador, Panama, and Russia, had significant differences in their p-values. Panama had a higher life stage, however, suggesting that house mice do not survive longer on urbanization alone. All the mice were found in provinces that would be considered urban areas. However, of the counties they were found, only one was from a county that would be an urban area. This would suggest that Panama has lesser levels of urbanization but has a higher average life stage for house mice. Both Ecuador and Russia had smaller life stages of mice, which matches the initial hypothesis that the US, with a higher population and therefore higher level of urbanization, has mice that live longer. Both Ecuador and Russia areas that the mice were collected in have mostly urban area populations but have smaller populations than most of the US states. These sets of data support the initial hypothesis.
The data does not strongly support the hypothesis that the house mice in the US will outlive those from other countries due to the level of urbanization. This is due to half of the country's data sets not supporting the hypothesis significantly. This experiment was limited by lack of samples from a multitude of countries. The 4 countries used to compare against the US had significantly less samples and therefore might not give the clearest answer for all of the country. Russia had the lowest number of samples, and they all came from the same 3 provinces in the entire country, which does not represent the entirety of the house mice that might live in the country well. A lot of samples were found from around the same areas as well, and did not give a spread of the country entirely. For Canada, all the samples came from countries that surrounded the same city, and almost all the Ecuador provinces were gathered around each other except for the Galapagos. This selection of samples does not represent the countries well either.
This study did bring about another question. What factor does affect the longevity of house mice if it is not urbanization? This could investigate environments and climate of areas that samples are found in, or the number of natural predators of house mice that are found in these areas. Determining this factor would help discover what external factors allow such small organisms to live longer than others. And while it was initially expected that environment and climate did not affect the longevity of the mice since urbanization was found not to be the major determining factor, this would bring about the possibility of these factors playing a major role.
For future experiments, it would be best to gather data from multiple databases to properly represent the life stages of mice in each of the countries. Not only would this likely increase the samples for the 4 countries compared in this study, but also allow comparison of more countries. Bringing larger numbers of samples would allow thorough examination of the life of these organisms and could help determine exactly what factor allows mice to live the longest they possibly can. And then these are our references that we used.